So we were learning the amino acids that will definitely be on these quiz. They're at least those that are 20 naturally occurring amino acids. Remember, they all have the common backbone, which I'm going to draw here. We've gone over all of these, of the non-polar ones. We also covered those that were aromatic and those that were positively charged are the basic ones. I think that if I remember right, we last left off with those that were going to be, I forgot, polar, and then the positively, no, and the negatively charged, right? Awesome. So let me bring this up so we don't skip anything. Okay. And yes, your first quiz is on Monday. The first exam is the Monday after that. And it's over chapters 1 through 4. Okay, let's see. Let's go. Yes. Sorry, I got way ahead of myself right here. Everything that one. Okay, so let's go with those that are probably negatively charged, or not probably negatively charged. We're gonna go with the ones that are negatively charged under physiological pH. And so the first up, is aspartate. Okay. Technically, it's aspartic acid until it loses its acidic hydrogen, and then, because it's a carboxylic acid, and it becomes aspartate. Its 3-letter code, does anyone want to venture a guess? ASP. Yes, and its 1-letter code is not an A, because that's alanine. It is D, as in day storms. And so then if we have the backbone, blah, 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 remember there's always that linking carbon except for glycine. And then technically, if we left it alone right here, what amino acid did I make? Alanine, okay? And so, but then all it is a carboxylic acid. But under physiological conditions, it's lost its hydrogen many times. So that's when it looks like that technically is aspartate. Aspartic acid, instead of the negative charge, it has the hydrogen on it. Okay. So unless you're given the pKa, or unless I specifically specify one or the other, you can draw it either as a carboxylic acid or the carboxylate. Does anyone know what's the other one that's negatively charged? Glutamate, or glutamic acid, okay? Hopefully you got the fear of God put into you. Doing the walk of shame. Oh my goodness. Okay, glutamate, glutamic acid. Once again, it's analogous to aspartate acid. So it's carboxylic acid. Many times under physiological pH, it's lost that terminal hydrogen. What is the 3-letter code for glutamate? GLU. This one's actually pretty easy to remember because its 1-letter code is E. So it spells glue. Okay. And just remember, aspartic acid is 1 letter removed from it. Okay, yes. So glue is glue. Be careful when you write, because you're going to find out that GLN and the U and the N can kind of look a lot alike for some of us when you write really fast. That's a different amino acid. And to make sure it's really obvious if it's a U versus an N, if you're only using the 3-letter code. It's very, very similar to aspartate. So we have that linking carbon, but all it is there's one other linking carbon and then the carboxylic acid or carboxylate. Well, once again, I drew it as a carboxylate, but you could also have it as a carboxylic acid, unless you were specifically told one way or the other. Or unless you were given pKa's and pH's and things like that. Those are the two that are negatively charged normally. Sometimes, so these are also called the acidic ones. Sometimes they also include tyrosine in this group, because if you remember, which we already did, because it's a phenol, which can lose its hydrogen. And then sometimes there's a third one that they like to include in this group, but I'm going to save it for the time being. That's cysteine. The reason why is I want to draw asparagine and glutamine right next to it. So these were the acidic ones. Now we're going to start doing the polars. And one of the polar amino acids is called asparagine. And I just like to draw it with alongside aspartic acid. And the 3-letter code for asparagine is ASN. And its 1-letter code is N. There's the backbone. And if you can draw aspartic acid, you can draw asparagine because they're identical, except instead of an carboxylic acid, it's an amide. It's supposed to be 2. It looks funny, but it's 2. NH2. It's just an amide. That's why it's a polar amino acid. So knowing what we know about aspartate and glutamate and asparagine, you can probably hypothesize what glutamine looks like. And glutamine's 3-letter code is GLN. That's why I try to always make it really obvious if it's a U versus an N. Does anyone know what its 1-letter code is? It makes no sense whatsoever. It's like, it's the unrelated stepchild of, sort of like tryptophan. Tryptophan's a W.
Y?
No. Q. It's Q. What one's Y? You've already done Y. Tyrosine. At least it has a Y in it. Yeah. No, this one's Q. I can't think of any way that's just one to memorize. I'll give you a moment. You could probably draw glutamine based off of the other 3. There used to be a whole section on the GRE that was kind of like this. We had to use logic. I love that section. I rocked that section. Got like 98 or 99 percentile on that section. What did they do? They did away with that section now. They think we don't need logic. They would ask you questions like, Zach needs to sit next to no one, but cannot sit next to, Sally, and Sally can't sit next to Ed. So what order must he sit, that kind of thing. And then little word games. Okay, so glutamine looks just like glutamate, except once again, instead of a carboxylic acid, it's an amide. That's it. Let me see. I want to make sure that I'm not, what ones I'm missing. I'm missing some more of the polar ones. Okay. So, yes, I know I have lecture, but thank you. All right, so for the next one, it's polar, but it's also sometimes grouped with the acidic because it has a hydrogen that can pop off. That's our good friend, one that we come to know and love, is cysteine. This is the only one where you definitely need to spell it correctly. Like if you misspelled the others, I can still make out what it was, then I give you the credit. The reason why is cysteine is similar, but not the same thing as cysteine, where if you miss that first E, it's cysteine, that's something different. So this one you have to make sure you spell it correctly. What's the 3-letter code for cysteine? Starts with a C. CYS. What's its 1-letter code? C. It's a simple one. See. Oh, stop doing that. Okay, here's the backbone. There's always a linking carbon. Does anyone know what functional group makes cysteine so special? It is. It's a thiol. Okay. Now, that hydrogen can pop off and it can be negatively charged S, because remember, sulfurs are better nucleophiles and better leaving groups than oxygen. And so then, what's it called whenever a thiol loses its hydrogen? Thiolate. Okay, so that's why it's really important. What's another thing that cysteines can do that none of the other amino acids can do? What can they form? Disulfide bonds. Okay, and when two cysteines come together and form a disulfide bond, that's called a cysteine. That's why you have to do this. So that's something special about cysteine. It's a great nucleophile, great leaving group. Make sure you only put one linking carbon, because when there's two linking carbons and it looks more like methionine, that's actually called homocysteine. And homocysteine is, we actually talk about that next semester in the metabolic pathways. Homocysteine is an amino acid. It's just not used to make protein. And it's used in metabolic pathways, and it can be an indicator of heart disease. Okay. Related to cysteine, or very similar, is serine. What's the three-letter code for serine? S-E-R. What's its one-letter code? S. Serines are fairly common. Okay, like pretty high up there with alanines and glycines and stuff. They're not very big. They look an awful lot like cysteine. There's the linking carbon, but what's the functional group hanging off of it instead of a thiol? Alcohol. That's it. Whoops, that's supposed to be an H. And once again, it can become an alkoxide, because it can lose that hydrogen speed, and then it's a really good nucleophile. So a lot of enzymes use serine or cysteine or threonine or tyrosine as nucleophiles for their the whatever functionality they're using. Because those are sulfurs and oxygen with lone pair electrons. Okay. Let me see. Next step is going to be threonine. Is threonine the last one that I have on my list? We've gone through all 20. We've done alanine, arginine, asparagine, aspartic acid, cysteine, glutamate, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine. That's one. Tryptophan, tyrosine, and valine. So yes, this is it. What's the three-letter code for threonine? THR. What's its one-letter code? T. That's why tyrosine can't be a T. Tyrosine is a Y. It's got the linking carbon. It's got another carbon. But what makes threonine so special? What functional group does it have? Alcohol. It has an alcohol, and there's only two carbons. The alcohol is right here. So it kind of looks like serine, but it has an extra carbon hanging off of it. Hang on. So it's got two carbons long, but remember the alcohol is off of the C beta. Because the carbon that's attached in the backbone is C alpha. Okay. Threonine and isoleucine are also special because not only do they have one chiral carbon, they've got two. See, the carbon that has the alcohol hanging off of it is also chiral. And isoleucine, the one that has the methyl group hanging off of it, is also chiral. Okay, so that's something that makes them special. Okay. Are there any questions? So we went over the 20 amino acids. Know them, come to love them, appreciate them, appreciate God's handiwork. I had at one time for pop, actually more than one time, for pop quizzes I've asked like, people like, which amino acid do you self-identify with, and like why? You're trying to get like some ideas and like experience. Like I've got some really interesting answers. So like this one girl like did a major like self-study using it. Like how she had a caustic attitude and all this, and so she was a strong acid. And I was like, what they didn't realize was I didn't know. I honestly did not know. And so as for fun, at the end of the day, at the end of the class, I was reading them, and I was having people guess. Like, so I was just randomly picking them up, and so it was like, I'm glycine 10 small, and so they'd say, oh, that's so and so, or whatever. But then this one is like, I have a horrible personality, I'm caustic, and it's like, we better not ask who it belongs to. You learn. You live, you learn. We're gonna have to go to therapy after this. So, I'm not a nice person. Okay, so the next few slides are just the amino acids in different forms. And so I'm spending more time on these, but you can see like what they all look like in different ways. So go, yay. Just want to take, this is a good table. It actually came from the old book, because it just has them all listed. All right. So all 20 are alpha amino acids. We've already discussed that. All of them have a chiral carbon, have serosine, except for glycine, and once again, isoleucine and threonine have two chiral centers, two chiral carbons. So it increases the different conformations that they can possibly have in different enantiomers. As I've mentioned, these are not the only amino acids out there. So I just throw up some of the examples here. You don't necessarily need to know these, at least not this semester. But just know that they're out there. And they're used for lots of different. There's also other ones. There's one called ornithine, which is named after birds, ornithology, which is important. We learned about ornithine next semester. Citrulline. But yes, there are lots of them. So these that are, they called it uncommon, but I guess it's kind of a, they are not used directly in translation. Typically these are modified post-translationally. So we have proline, and there's one called hydroxyproline, where that middle carbon off of the home plate looking thing is hydroxylate, has an alcohol on it. And likewise, there's lysine and there's hydroxylysine. And these are really, really important in collagen. So next chapter, I think it's next chapter, I've got pictures of it, especially hydroxyproline in collagen. Like 1/3 of the amino acids in collagen are prolines or hydroxyprolines. And that's one thing we already talked about the importance that proline is permanently kinked, it's rigid. And then the hydroxyproline can, not only is it rigid, but it can still hydrogen bond between the different strands of collagen. Okay? Then there's thyroxine, which is a derivative of tyrosine. This is an example, and it's found in the thyroid gland. It's adenylated. Okay. I would never ask you to draw those out, except for the original parent amino acid. There are amino acids and neurotransmitters. We were talking about this. Tryptophans used to make serotonin. There are those that are the dopaminergic ones, which come from tyrosine. So we can go on. So dopaminergic, sorry, epinephrine. What's another term for epinephrine? Epinephrine is also, you're running from a bear. What kicks in? Adrenaline. Which I always put a little disclaimer here. You're going to hear this disclaimer over and over and over again until next year. If you run from a bear, you run downhill, don't run uphill. Because bears are bottom heavy, and so they have problems running downhill and can fall. I don't know if it's like a factor that has stuck with me all these years. It's like you don't climb a tree when you run from a bear.
But Floyd is all flat, so.
Yeah, then you're in trouble. But if you're back home, my home, then run downstairs. Just like I usually have lots of disclaimers. This isn't forensic science, but I have lots of disclaimers with respect to firearms that I use. So, yeah. We actually go over this kind of stuff next semester. So we go over student time permitting. Usually neurotransmitter metabolic pathways was the very last chapter that we covered. in biochem 2. Okay, we've actually already discussed this. This is just it's written out explicitly so that way you take note. So this is just the backbone. It's Witter ion. Whenever it's in its isoelectric, the Witter ionic state, it has a 0 net charge. If the pH goes low enough, goes below the pKa of this carboxylic acid, then it's going to have a +1 charge overall because the amino group is positively charged and so is the carboxylic acid. Now, that's assuming that the R group doesn't have like something that can be charged as well. So this is why this is true for like alanine, glycine, valine, things like that. If you go to really high pHs, not only has this hydrogen fallen off, but so has the one that's gone above, whenever you're above the pKa for this one. And now it has an overall -1 charge. Once again, there are some other amino acids that their R group also has pKa's, and so therefore they can either add positive charges or negative charges. Okay, and so that's what this is trying to show you. In general, most of the pKa's for the carboxylic acid is right around a 2. Like 2 to 3, just like the pKa's for the amino group is usually 9 to 10. I would not ask you to know the exact numbers. Okay. But I would expect you to know the range. All right. So this is more acidic than what you'd expect. Because if you remember back to Gen Chem, acetic acid, which is vinegar, that's the one they like to use in almost every examples for acids and pKa's. It's 4.76, or almost 5. Whereas the average pKa for the carboxylic acid on an amino acid, on the alpha amino acid, is very low compared. And the reason why is because of the fact that that amino group is there in the conformance of the Witter ion. Okay? This is where you have to kind of review Gen Chem. These are titration curves. Now, this is a titration curve for something that has an R group that does not have a hydrogen that pops on and off. This is where it gets kind of tricky. To read these, this is pH. This is as you add base. You could also do it as you add acid, but then if you do it as you add acid, the pH is going to start higher and end up lower. It's going to go that other direction, okay? So usually they just do it with respect to base. You will have these isoelectric points, isobestic points, points of inflection. That's probably better. That's the term I'm needing. The point where the entire molecule overall is neutral is called the isoelectric point. It's a pi. Each amino acid has its characteristic pi. Each one of these other inflection points is an individual pKa. So the problem becomes, if we have something like glutamate, for example, which has glutamic acid, instead of going to 2.0, you've got to go all the way to 3.0, because it's got 3 hydrogens that can pop on and off. It's got the one for its side chain, as well as the one for the carboxylic acid here, as well as the amino acid. Okay, so that's why some people, and there are some suggested problems, I think, if I remember right, where you have to draw some of the titration curves, where you're given the numbers, but you have to draw the titration curve, or you're given the titration curve and you got to figure out like a certain pH what the charge would be. You have to realize that the total number of, well, that is huge. The total number of moles of base added has to equal the total number of moles of hydrogens that can pop off. Every amino acid is going to go up to 2, but some of them will go up to 3. It may take some time to sit there and figure, to think it through. It's because it's been a while. We can. This is to show you, if you have glycine, which only has 2, the pi is going to be average. It's really simple. If you have something like histidine, where you've got 3, it is not as simple. Okay. I would never ask you to memorize this. so please do not memorize this. But if I gave you this, I could see where I could ask you to draw the titration curve. Or if I gave you this and a titration curve, or I gave you this, you didn't even need the titration curve, and I gave you a pH, I would expect you to figure out the charge. So for example, if we look, let's just pick one of these. Oh, first of all, I just want to point out, see how roughly the pKa for all of these is right around 2. All of these are like 9 to 10, give or take a little bit. And these are the PIs. If we look at, it doesn't matter which one of these that we do. Let's say, whoops, at pH 8. And we picked one of these out. Let's just look at threonine. Which technically it's got a hydrogen that can lose it, but it's at such a high pH that it loses it that it's physiologically not relevant. Okay, because it is an alcohol. But at pH 8, trying to figure out which ones still have their hydrogens or not. Okay. So some people just need to draw the little titration curve. Remember, it's going to look like this, roughly, or something like that. Where this is the pKa1, pKa2, and right there, so it would be the PI. Something similar to that. Okay, but if we look, all you need to do is at pH 8, will this one right here have the hydrogen on it, or will it be off? The pH is higher than the pKa. So what will that carboxylic acid look like? Will it be a carboxylic acid, or will it be a carboxylate? It's a carboxylate, so it's going to look like this. Okay, then we go over here. Can you guys see? Yeah, it does show up. All right, in it at pH 8, you haven't reached the pKa yet of the NH3 group. So will the hydrogen still be on it, or will it have lost its hydrogen?
Still be on there.
It's still on there, so it really does look like NH3 plus. You have to go above 10.43 before you really lose. Technically at 10.43 you lost half of them, so it's a half of a charge. But yeah, so now all you do is you just add these up. And so at pH 8 you have a -1 and a +1, so it's neutral. Okay.
We'll go for the other alcohol group.
This one, I don't know what the pKa is of the other alcohol group, but it's at such a high pH that it's physiologically not relevant. If we look at the other ones, see this one now has the side chains for the ones that can lose it. They don't have serines on here, which I don't know what serine, I wouldn't say serines is pretty high too, but it is possible in serine to lose it. So if we look at aspartic acid, for example, this is one where if we were at pH 8, it would have lost the hydrogen here, so that's a negative. It still has a hydrogen there, so that's a positive, but it's lost this hydrogen there because this is a carboxylic acid. This time you have to draw out what the side chain is. So it's got a -1, +1, and -1, so it would look -1. Okay, now where it gets tricky, something we haven't really talked about yet, is what happens when you have more than one amino acid stuck together in a peptide. So hopefully we have enough time for me to explain how to do that later, because I don't want to jump ahead since we haven't done that yet. But because what's going to happen is you're going to lose some of the charges because of the fact that they're involved in bonds between the amino acids. Okay, but once again, you may need to sit down and just kind of think back through Chem. Okay, we're going to skip electrophoresis for right now. It's covered. I got it in here twice in these PowerPoints. So. All right. Actually, maybe it'll get, since I skipped electrophoresis right now, maybe cover like what happens when I figure out the pKa's. So now we're going to start to make proteins. Emil Fisher, the good old guy who came up with the Fisher projections, is the one who proposed how the amino acids came together, and he was correct, by the way. Okay, they call them peptide bonds. They call a string of amino acids in a row a polypeptide. So a big protein complex may have more than one polypeptide. They use the same term, whether it's multiple amino acids in a row or multiple protein chains coming together, so it kind of gets redundant. That's a peptide. And so it's an amide. So what happens is you have the carboxylic acid of one amino acid and the amino group of another amino acid coming together, smacking inside the ribosome to make an amide and giving off water. Okay, so what that looks like would be right here, a little cartoon. Many times you skip steps, unfortunately. In biochemistry, you're gonna find that out a lot. But we have this, right now it's shown as a positive charge, but inside the ribosome, this would have lone pair electrons. So essentially what we're gonna be doing is it smacks that carbon, the carbonyl group, you form a tetrahedral intermediate, you collapse the tetrahedral intermediate and it kicks off a water, and that's when you form the peptide bond. So if you notice now, whenever we go to figure out the pH overall of this dipeptide, we've lost one carboxylic acid and one amino group. So people forget that whenever they start to figure out the charges. And so then we have to really look at the R groups. So for a big polypeptide, you only have one free amino end and one free carboxylic acid end. Then you just have to look at all the R's. Okay, so this is what it looks like. Don't be surprised if I don't have you draw out peptide bonds at some point in time. Next chapter we go into greater detail, like what makes peptide bonds so special. There's always a directionality, just like with DNA and RNA. You probably learned it in genetics or biology. What's the direction for DNA or RNA? They always talk about going from one end to the other end. 5' to 3', 5' to 3'. Well, for proteins, they do the same thing, but we don't have 5s and 3s, because those are numbers you should now recognize that the 5 and the 3 comes from organic chemistry's locant for that position. Here, they also use locants, but they use letter locants. So it's the N terminus, the amino end, to the C terminus, the carboxylic acid end. Okay, so there's always the amino, the N terminus, to the C terminus, and the C, and the C, and the C. Okay. So that's the directionality for it, the peptide bond. Just like, let's say, for example, if they give you a peptide sequence, what this means, phi, ala, gly, asp. That literally, that's supposed to be an E. That literally means, this is the N terminus, probably to make it more obvious. They don't have to put lines there, but I'm just trying to make it more obvious to fit like what I was there before. The N terminus is the phenylalanine, the C terminus is the aspartate. If we wanted to figure out the charges, say once again at pH 8, this is what I suggest that you do. Draw, this is the amino end. So I'll draw it this way to make it. This is the carboxylic acid end. And then you have to think about each R group. The R group of phenylalanine doesn't have a pKa, because it doesn't have the hydrogen that pops off. Same thing with alanine and glycine. However, aspartate does. It has another carboxylic acid. So at pH 8, now we have, once again, you'd have to look back at that table, but we already did this aspartate. We have a +1 charge right here. Oh, I didn't mean to do that. A +1 from that end, and a -1 from this end. And we have a -1 for that carboxylic acid from the asp. So we just add this all together. So overall, this polypeptide would overall have a -1 charge at pH 8. That's what I mean, because you have to realize that the amino group from alanine and the carboxyl group from phenylalanine, the backbone, doesn't count anymore because they're in amides. People usually have at least a handful of people that forget that. And so they make it much more difficult than what it really is. Okay. That was at pH 8. Just pH 8. Because obviously if you go to different pHs, then it can change things. Okay. Actually, I'm not going to spend a lot of time on this. If it's just two amino acids called a dipeptide, then you just try peptides. Usually anything beyond 3, they just call it a polypeptide. Technically speaking, a protein, and it's not set in stone. Like if a protein weighed, or if the polypeptide weighed 4,950 grams per mole, it's not going to get kicked out of the guild of being proteins. You know, it's kind of relative. It's a gray gray thing. But many times they would just use that term polypeptide and protein interchangeably. But essentially, once you get to larger sizes, they're called proteins. One thing that actually no one specifically told me, what units do they usually use when they talk about the molecular weights of proteins? Does anyone know? Like in your papers and stuff as you start reading about your enzymes, very rarely do they give it to you in grams per mole, which is what you're used to in organic chemistry and gen chem. Does anyone know what's the unit they give for proteins in biology and biochemistry? It's a Dalton. They'll say Daltons or kilodaltons, which is just 1,000 Daltons. And 1 Dalton is equal to 1 gram per mole. No one ever specifically told me that. So, I don't know what a Dalton is. Good for him, it's named after him, but what is it? That's how you can actually relate it back to the periodic table as well. I don't even know if it still is that way in the book or not. Okay, so now let's start looking at some of the important small peptides. So these technically would not be proteins because they're very tiny. So this is when I was asking when someone, because when I first started, the big push was to get away from the sugary nasty drink. No, I shouldn't say that. Not moderation. That's why I said moderation. Same thing with juices. But people would have some Diet Coke or something like that, and you can't even use Diet Pepsi anymore because Diet Pepsi no longer uses aspartame. But on it, anything that uses aspartame, which does anyone know what's the brand name for aspartame? Like it's the little packet, it's equal, like the little blue packets. But this is aspartame. First of all, there are people that are going to be allergic to it. You can be allergic to anything, essentially. But all of it is L-aspartyl L-phenylalanine. Notice the YL is just like from organic chemistry. It just means it's attached. It tells you from the N terminus to the C terminus, the only thing that makes it different than what your body would have naturally in it is there's an extra methyl on the end. That's it. There are some people that are allergic to it. Yes, you can have too many problems with it. Small amounts, you're probably fine. Okay, unless your doctor tells you otherwise. But there's this big evil conspiracy. I don't know. There's lots that you can say with it. But in reality, it's not going to kill you as long as you're not allergic to it, or as long as you're not a phenylketonuria, which we'll talk about in a moment, because it's got phenylalanine. Next to it, I've got beta-alanine L-histidine. Notice it says beta-alanine because this is when the carbon, the amino group has been shifted from the alpha carbon to the beta carbon. Beta-alanine L-histidine, which is carnosine. Carnosine is actually a muscle supplement. Okay. Or can be used as a muscle supplement. Don't confuse it with carnitine, which is slightly different. At some point, I haven't checked to see if that link still works, unfortunately. But there are some people that are looking at it to try to use it as a possible supplement for certain types of autism, for improving motor skills and things like that. But okay. Other examples: oxytocin and vasopressin, which I talk about these again next semester. Notice they're using the 3-letter code. They are almost identical, oxytocin and vasopressin. What's one difference between the two? Pardon? There's a phenylalanine and isoleucine. There's one other change. Arginine and a leucine, okay? This change here is typically not considered huge, right? Because these are both nonpolar. This one is aromatic. This one's a branched chain amino acid, so there is a change, but it's not like earth-shattering. What kind of amino acid is leucine? Is it polar or nonpolar? It's nonpolar. It's another nonpolar branched chain amino acid. What kind of amino acid is arginine? Basic. It's positively charged usually. That's the big difference between these such small changes. What is oxytocin? I mean, these are both hormones, but what does oxytocin do in your body? Who typically has more oxytocin, men or women? Women. Okay, so why? What does it do? It does. It helps with muscle contractions during labor. That's one. That's a big one. In fact, Pitocin is the brand name that they give women to induce labor. Okay, and what else does it do? Think about it's very similar to muscle contractions in women and babies, and it also helps with production of milk, pushing the milk out. Okay, versus vasopressin. What does vasopressin do? First of all, who has vasopressin? Which men, by the way, do have oxytocin. Oxytocin is also believed to be involved in helping with the bond between mother and child and possibly even father and child. Now, there's the big push, when your baby, even for men, when the baby's first born, that you take the shirt off and you're supposed to hold the baby to your chest and things. And I did. For two reasons. One is supposed to help with the bonding of the child, but two is that's newborns don't have any way to regulate their heat. And the best way to regulate their heat is actually with an adult's body heat. Okay, but what about vasopressin? Does everybody have vasopressin in it, in their body? Yes, but what does it do? It does help with blood pressure, yes. And so something completely different. Oh, whoops. I didn't realize I had these on two different sides. What it does is it helps with the water uptake. It's an anti-diuretic. So therefore, if it causes water to be uptaken, what is the word? Is that the right word? I can't see. That just sounds weird. If it causes the kidney to take in the water, then it acts as an anti-diuretic, it's going to increase your blood pressure. Okay. Uptaken. Don't know. Crazy English language. Okay, here we got time. Good. And as I mentioned, PKU, and I haven't checked to make sure to see if the link still works. They may have broken the link. PKU is phenylketonuria. And so people who suffer from it many times are called phenylketonurics. We actually talk about this more related to what we do next semester. I apologize that here in the room that this is phenyl acetate, which you may remember from organic. It was one of the compounds I used to always point out whenever I did esters and organic chemistry. Okay, and this one right here is tyrosine. So normally, people have, this is an old term, oops. This transaminase, which ASC means it's an enzyme, the more correct term is called aminotransferase. Which we talk a lot about aminotransferases next semester. But yes, next semester we do this kind of thing all the time. Normally, what happens is your body, when we have phenylalanine, what we do is we can break it down two different ways. The way that we often do it is we take phenylalanine, excess phenylalanine, and we convert it to tyrosine. Tyrosine can be used for lots of different things. Okay. By the enzyme phenylalanine hydroxylase. So the only difference, and I suggest that you learn your enzyme family names. Hydroxylases are enzymes that add alcohols. And so that's why, if you know the enzyme name, it tells you a lot about its activity for metabolic pathways. But that literally means that it's an enzyme that adds in a hydroxyl group to phenylalanine, so it converts it to tyrosine. Our body uses tyrosine for lots of things. The problem is people who have PKU, genetic PKU, their phenylalanine hydroxylase is deficient. It doesn't work correctly. And so your body has to get rid of this. This would be bad to always have around. So it uses an aminotransferase to remove the amino group to turn it into what's called an alpha keto acid. Alpha keto acids are also really important in your body that we talk about next semester. But this one we don't use frequently, okay? This is not the best way to get rid of it. And so it turns it to phenylpyruvate. It looks like pyruvate, so it's got a phenyl group on it. It's not done. There's a dehydrogenase here that will go ahead and it turns it into phenolactate, which is related to lactic acid, which then decarboxylase removes that carboxyl group and turns it into phenylacetate. Phenylacetate is bad. Okay, you're gonna find out a lot of these carboxylic acids in the end, if your body can't get rid of it good enough, then they're bad. They cause osmotic problems. They also affect pH because they're acids. So what happens with PKU is as the child, because usually, now they test for PKU in Florida, it's required the moment child is born. Okay. And most states, I'm pretty sure that's common through all 50 states in DC. They test for it because the problem is you have to go on a restricted diet immediately. They are not allowed to have, they cannot have phenylalanine. They have to have as little as possible because they can't get rid of it. And so what happens is it causes osmotic problems for them. And so it accumulates within your brain. Water comes in to try to get rid of it, which it can't easily. It expands, and so many times they end up being severely retarded or they die. Okay? And it's also believed to affect pyruvate acetyl CoA, which you'll find out next semester, is the first step to try to get into the citric acid cycle for energy. Okay. Are there any questions? Because this is like an awesome place to leave off.